Hello everyone, welcome to Bioscient once again. I hope you all are doing well. The exams are coming, so let's be focused. And today I am going to continue the chapter which I have started earlier. That's the biotechnology chapter, which is very important chapter. Okay, so in the previous lecture of mine, I have delivered about the enzymes used in biotechnology. Those who are seeing the video for the first time, they can subscribe the channel, and the link of the previous video will be there in the description box. Let's get started. So, one of the important component or the prerequisite for performing any experiment in biotechnology, we need vectors, which is an important tool for biotechnology. So first we have dealt with the enzymes. Now I am going to discuss about the vectors. Vectors means what? Vector means the one who or which carries something. I hope you know this by now, that some of the bacteria are blessed with this vector called plasmid. What are those plasmids doing in bacteria? Actually, they are like resistance power, like you have immunity, right? Similarly, some of the bacteria are resistant towards certain antibiotics. So those special kind of property which is governed by some genes are actually present in the plasmid, which is not present in all the bacteria, of course. So the plasmid may possess some important genes. Now that might be beneficial for the bacteria, but it is not for us. Don't you agree? Because the bacteria, if it becomes antibiotic resistant, then how will we treat it? So it will be definitely a difficult task for us. Now what if we are doing something which reduces the harmful effect of the bacteria, as well as it's doing something favorable for us? It's like blessing in disguise, isn't it? So we are going to use that kind of vector which will actually, we are going to remove the harmful genes from them, and of course they are going to infect. So while infecting the other cell, it will be carrying our gene of desire, isn't it? So that is how it is going to be useful for making products which are important for the human welfare. Now, if we are going to select any vector, then the vector should have these property. I think you know that the eligibility criteria. So a vector which is important for selection in the process of biotechnology, it should have the following characteristic or property. Of course, it should have a ORI site. What does that mean? It's origin of replication. Origin of replication. Why is it important? Why is it important? Of course, we want our gene to multiply, or it will produce more and more protein, of course. So if the plasmid will have this property of replication, then when the bacterial gene or whatever gene they have, it is going to multiply, then of course it will be helping us to make multiply the gene of our interest as well. Okay, so I hope you are getting it. The another property which is important for a vector is replication protein. What does that mean? Of course, there will be an enzyme required for replicating the gene. So it should have a protein or the site where the replication will begin, or so origin of replication, and then ROP has an equal importance because DNA polymerase will be helping in the replication of that particular DNA. Now, an ideal vector should have selectable markers. What does that mean? If your product is ready, how will you distinguish like now the product is transformant or the recombinant, so it should be distinguished from the original one. So the ideal vector should have selectable markers. Okay, so antibiotic resistance gene could be one of the selectable marker, like we are going to discuss about the one of the important vector which will have ampicillin or tetracycline as the antibiotic resistant gene. Now, somewhere in some of the plasmid, you won't find any kind of antibiotic resistant gene. Rather than, we can use this property that's called the enzyme producing property, which can also be used as selectable markers. I will be telling you the example of this. Okay, once again I'll repeat. The property of an ideal vector, ORI, ROP, selectable markers. Now, as I have discussed in the previous class, every vector should have a restriction site. What does that mean? Restriction site means where the restriction enzyme will act, and the restriction enzyme will cut the plasmid so that we can insert the desirable gene into it, isn't it? Now, if just imagine if it has the same restriction site at multiple sites. Now, if you're using a restriction enzyme there, your plasmid or whatever vector you have taken, it will be cut into many pieces. So this purpose will not be solved. So an ideal vector should have what? It should have one restriction site with one restriction enzyme can work. That's the best part of it. So that it would be easier for a person to work upon, so that it will make the plasmid which is circular into a linear one. Now, one of the important prerequisite for the vector is it should have a small size as well. Why is it so? It would be easier to handle with. Okay, so here the NEET aspirant, you must know that what are the important vector, and you will get the question line by line from the NCERT. Okay, so you must have seen a diagram in the chapter of biotechnology of a very famous vector. This is the first artificially designed vector, that is PBR322. PBR322, the nomenclature goes like, it is P for plasmid and B is for Bolivar and R for Rodriguez. These were the two scientists which has designed this plasmid. Okay. As I told you, it should have a origin of replication, which it ideally have. Then it should have replication protein. Then the antibiotic resistant gene. This question was asked that the PBR322 has which enzyme resistant gene? So it has ampicillin and the tetracycline resistant gene. Okay, these two sites can be used for transfer of your desired DNA. And then there are multiple sites, restriction sites. So basically there are 8 restriction sites. How many? If you see 1, 2, 3, 4, 5, 6, 7, 8. It has 8 restriction site, means it can be used with the 8 restriction enzymes. So how do we name these restriction enzymes or the restriction sites? I have already told you in the previous video, like E. coli was extracted from E. coli, and this was the strain number 1, and accordingly the nomenclature is being given. Okay, so coming back to the first is the PBR322. Then we have got PUC. This is PUC family, the another kind of plasmid where the selectable marker is LAC-ZZ. Okay, which produces the enzyme. I would like to know from you which enzyme is encoded by the LAC-Z gene. Okay, now we have got TI plasmid. This is very, very important. The source of this plasmid. You must have heard this biotechnology chapter is incomplete without knowing the name of Agarobacterium tumefaciens. Agarobacterium tumefaciens has a special type of plasmid called TI plasmid, which has a property of inducing tumor in the plant cell. Okay, so basically it carries genes which induces tumor. Now we can use this plasmid by protecting the plant from getting tumor and adding your desired gene so that a plant can become insect resistant, which is the topic of the chapter, where you will see how we have made the GMCs or GMOs. GMC, genetically modified crops, the examples would be BT cotton, BT brinjal, that I will be discussing in the coming lectures. So, TI plasmid, the way it has TI plasmid tumor inducing, there is another RI plasmid as well, root inducing plasmid, and then we have got bacteriophages. Bacteriophages, since lower classes you must have studied that. Bacteriophages means what? These are the viruses which infect bacteria, isn't it? So this bacteriophages will be transferring the DNA or the genetic material into the bacteria. So while transferring their genetic material, you might use them as your vector to take your desired gene, isn't it? Similarly, during Human Genome Project, two important vectors were used: YAC, that is Yeast Artificial Chromosome, Yeast Artificial Chromosome. You just need to remember the names of these vectors. And BAC is Bacterial Artificial Chromosome. Now, another two important vector which was designed, the cos site plus plasmid. Cos sites plus plasmid will give you cosmids, and bacteriophage plus plasmid is phasemid. Okay, so we are through with the another important tool of biotechnology, that's the vectors, and there are many more things yet to come. So stay tuned, and if you have any suggestion you can write in the comment box. Till then, stay happy and keep studying. Thank you so much.